This is D1.2 Protein Synthesis from the new IB Biology curriculum. Our two essential questions are, how does a cell produce a sequence of amino acids from a sequence of DNA bases? And how is the reliability of protein synthesis ensured? We are going to focus on two processes, transcription and translation. This is why you have DNA. The purpose of DNA is to provide a code that codes for proteins. And we're going to see the production of proteins happens through two steps. First up, DNA is transcribed into mRNA. This process of transcription occurs in the nucleus in eukaryotes. That mRNA is a copy of a part of the DNA. It's coding a gene and making a copy of that called messenger RNA. Now that messenger RNA is then going to exit the nucleus through a nuclear pore into the cytoplasm, where that code is going to be coded into a protein. Now, a protein is made up of a chain of amino acids called a polypeptide that then folds into its 3D shape, which we then call a protein. This process of translation always occurs in the cytoplasm, whereas transcription occurs in the nucleus in eukaryotes. But remember, prokaryotes don't have a nucleus, and so it actually occurs in the cytoplasm as well in prokaryotes. Okay, you might visualize protein synthesis like this. We know that DNA is double-stranded. This template strand, through complementary base pairing, is going to code for that mRNA. Every 3 bases on this mRNA copy are going to code for an amino acid. That chain of amino acids, called a polypeptide, then folds into our protein. So let's break down these steps one by one. Transcription is the synthesis of RNA using DNA as a template. So this DNA you have one master copy of. It stays safe in the nucleus. It never leaves. But in order for these proteins to be made, we're going to have these sections of mRNA that are produced based on complementary base pairing with the template strand of DNA. Because RNA is single-stranded, transcription only occurs along one of the 2 strands of DNA, called the template strand, and we can transcribe genes repeatedly to provide as many copies of a base sequence as needed. That's going to be called gene expression, where which actual genes get turned into proteins can be controlled by which genes have an mRNA copy made of them. Those mRNA copies can be made repeatedly depending on how much of that protein is needed in that particular cell. Now remember, every cell in your body, other than a few exceptions, has your entire set of DNA. But not every part of your DNA is going to get coded into proteins. Maybe you're a muscle cell. You're not going to need the protein for insulin. Insulin is produced by pancreatic beta cells. And so if you're a muscle cell, you're never going to make insulin. And so that part of the DNA, you have the code for insulin, but it's never going to be made. So only the portions of the genome that are specific to the function of that particular cell are going to be made into mRNA, and then the mRNA is going to code and be translated into that protein. So let's run through transcription. Whereas in DNA replication, we saw that there are multiple enzymes involved that you need to be familiar with. Here in transcription, we've got this one-stop shop, RNA polymerase. Just like it sounds, remember that we are going from a monomer to a polymer. When we take these monomers of free RNA nucleotides and we join them together into a polymer, that is going to be done by a polymerase enzyme. And this one is specifically making an RNA polymer, as opposed to what we saw in DNA replication, we were using DNA polymerase. So we're not making DNA anymore, so we're using RNA polymerase. What does it do? First, it's going to bind to a site on the DNA at the start of the gene that is being transcribed. It then unwinds the DNA double helix and separates it into 2 strands. It's then going to unwind the DNA double helix and separate it into 2 single strands. RNA polymerase is then going to move along the template strand, and it's going to position RNA nucleotides on the template strand with bases that are complementary to those of the template. So here you can see the template strand is in blue. Okay, RNA polymerase is this big enzyme in kind of this light gray, purpley color. And so it is controlling adding these nucleotides one by one in a 5' to 3' direction, all the way down using complementary base pairing rules. As those RNA nucleotides are positioned, RNA polymerase is going to link those RNA nucleotides together by covalent sugar phosphate bonds. And that's going to form a continuous strand of RNA. So sugar phosphate bonds between nucleotides, just like we saw in DNA. Then it's responsible for detaching the assembled RNA from the template strand. The DNA double helix can reform, and this can be made an unlimited number of times. These copies can be made that exit the nucleus that allows for DNA, the master plan, to stay safe. So, of our 2 strands of DNA, one strand is our template strand. This is going to be the strand that is used to make those complementary base pairs with those RNA nucleotides. The other strand is called the sense strand. The sense strand is non-coding. It's just matching up with the template strand. This strand is going to be almost identical to the RNA produced, with the exception in the base sequence of a uracil wherever there would have been a thymine in the sense strand. So these would be the same all the way through, except anywhere there would have been a thymine in DNA, now there's a uracil in RNA. DNA is incredibly stable. This is why DNA has lasted as the genetic material in all living things, because it's incredibly stable. These mRNA copies can be made an unlimited number of times without the DNA base sequence changing. In somatic cells, those body cells that don't divide, such sequences are conserved through the life of a cell. And so your DNA is there for the entire life of the cell, and just mRNA copies are made as needed of certain genes. If mutations were common, then frequently used templates would accumulate mutations and RNA copies would contain more and more errors. But mutations are incredibly rare. So this process is called gene expression, the process by which information carried by a gene has observable effects on an organism. The function of most genes is to specify the sequence of amino acids in a particular polypeptide. The polypeptide is a chain of amino acids that we then call a protein when it's folded into its 3D shape. It is the proteins produced that directly or indirectly determine the observable characteristics of an individual. Transcription is the first stage in gene expression, and it's the key stage at which genes can be switched on and off. So whether they get transcribed into mRNA or not is gene expression. This is what we talked about earlier. Only certain cells are going to need certain genes. Depending on the cell's job, depending on the cell stage in its cell cycle, there are going to be different genes that are needed. Some genes may never be switched on during the life of a cell. Like we mentioned earlier, the gene for making insulin in pancreatic beta cells will only be needed in pancreatic beta cells. And so while all somatic cells have the code, it's never going to be transcribed. There are also genes that are always going to be expressed because the proteins they code for are always required. For example, genes that code for proteins required for cell respiration. Another example of genes that are only switched on sometimes are going to be all of those genes we just studied in DNA replication. We're only going to need to have those genes during the stage in the life cycle where DNA is being replicated. Enzymes like DNA polymerase III, DNA helicase that we just studied are only going to need to be produced at certain times during the cell's cell cycle, and so it wouldn't make sense to produce them all the time. That would be wasteful. Let's pivot now to translation. Translation is the synthesis of polypeptides from mRNA. So now that mRNA has been made by transcription in the nucleus, that mRNA is going to exit through a nuclear pore into the cytoplasm, where it will be met by a ribosome. To make a specific polypeptide, amino acids must be linked together in the correct sequence. And that sequence is what is coded for by DNA. The purpose of having DNA is to give instructions about what order these amino acids should be placed in. A typical polypeptide is a chain of hundreds of these amino acids, of which there are 20. Translation is defined as the base sequence of mRNA that gets translated into a polypeptide. So it goes from mRNA to polypeptide. Again, it happens in the cytoplasm. RNA produced in the nucleus is going to pass out to the cytoplasm via a nuclear pore. Distances are shorter in prokaryotes, but transcription and translation happen in different parts of the cell. So even though in prokaryotes, because they don't have a nucleus, transcription will have to happen in the cytoplasm, but there are different parts of the cell in which transcription and translation occur. The RNA with the information for making a polypeptide travels from one place to another, and so it's known as mRNA or messenger RNA, copying the message from the DNA and taking that copy to the ribosome. Now, there's a few terms you'll need to know in order to make sense of this process. First step, the mRNA, which is that copy of the DNA, every 3 bases on the mRNA is called a codon. These 3 bases are going to code for 1 amino acid. They are going to be complementary to what's called the anticodon, which are 3 bases on a tRNA molecule. Now, tRNA, when drawn out, sort of looks like a T. The T stands for transfer, because its job is to transfer a specific amino acid to the ribosome, which is going to be building the polypeptide. So the sequence of the amino acids is determined by the sequence of bases in the codon, because they are only going to match up with a specific complementary anticodon, and that anticodon is going to carry a specific amino acid. Here is our mRNA. Again, you can see every 3 bases is 1 codon. We'll always have one specific start codon, which is the codon AUG, which will always code for the amino acid methionine. And there are 3 different codons that are our stop codons. When this codon is reached, the ribosome will dissociate from the mRNA, and the mRNA can be used again for a few more times. The mRNA can get broken down if it becomes more damaged, or if more copies of the polypeptide it codes for are not required. So here's that tRNA. So when drawn out in 2D form, it looks sort of like the letter T. The important part is that these 3 bases form the anticodon. This is more of a 3D rendering. It looks to me like one of those little space guns, like a pew pew. Okay, here's our anticodon, those 3 bases of RNA that are going to be complementary to our codon on the mRNA. And at this end, we have our amino acid attachment site. Remember, only a specific amino acid of the 20 will attach to any given tRNA based on its anticodon. Each type of tRNA is going to be activated by having an amino acid added to it using a specific enzyme. Then we'll talk about a ribosome. Ribosomes are made up of 2 subunits, a large and a small. I think of them sort of like the buns of a hamburger. The top bun is the large subunit, and it's going to have 3 binding sites for those tRNA molecules. The small subunit is like the bottom bun, and this is what's attached to the mRNA. This ribosome complex will slide along the mRNA, and as it slides, new tRNAs will enter into the ribosome. If the codon and the anticodon match through complementary base pairing, then this amino acid will get dropped off. This growing polypeptide will form a peptide bond with this new amino acid. The whole ribosome will shift over 1 codon, and this process will continue. So we'll walk through that. But you should know that the bonds formed between amino acids are peptide bonds, hence we call it a polypeptide when we have a chain of amino acids. So the reason that this works is because of complementary base pairing. This ensures the sequence of amino acids is correct. We're always going to have A bond with U and C bond with G. This is mRNA and tRNA, so we'll always have the uracil base instead of a thymine. The thymines are back with the DNA in the nucleus. Now remember that every 3 bases on the mRNA form a codon, and they're going to code for a specific amino acid. This is an example of an amino acid table that tells us which amino acid is coded for by any particular 3-base sequence. So, for example, we talked about AUG being the first codon. So to use this type of table, we'd first look for the first base over here on the left side, and here's A. The second base is across the top, so here's U. So A and U were somewhere in these 4 when they match up, and then we look over here for the third base. So G would be this bottom one, and sure enough, there's methionine, our amino acid, which is always going to be our first amino acid. Now it can appear later in the sequence as well, but it will always be the first one. Now there are 20 different amino acids, And so if there was just a one base to one amino acid, we'd only be able to code for 4. And if there were 2 bases that coded for an amino acid, we'd only be able to code for 16 amino acids, which is why we have to have 3 bases. That way we have 64 different options as seen here. But because there are only 20 amino acids, there are going to be several codons that code for multiple amino acids. We can have between 1 and 6 codons that code for any given amino acid. Now remember I mentioned that AUG is our start codon, but we also have our 3 special codons that don't code for an amino acid. If either UAA, UAG, or UGA is the codon, that is going to cause a stop. We are not going to have an amino acid bind if that codon is reached. Instead, that dissociation will occur of the ribosome and the polypeptide will be completed. 2 notable features of the genetic code are degeneracy and universality. So we say the code is degenerate because different codons can code for the same amino acid. For example, the codons GUU and GUC both code for the amino acid valine. So let's confirm GUU valine, GUC valine. So it's degenerate because multiple codons can code for the same amino acid. The code is also universal because it is used by all living organisms and viruses with only very minor changes in some cases. So this same code, whether it's a chimpanzee or a frog or a bacteria, the codon CUU will always code for the amino acid leucine. This is why with biotechnology, we can cut and paste DNA between species to make proteins that are coded for by the DNA of that first species. And this is how we make human insulin for patients that have diabetes. We take that DNA that codes for human insulin and we use the mechanisms in a bacteria to make human insulin. It's not bacterial insulin, it's human insulin because we've taken that DNA and that bacteria uses the same mechanism, and so it reads the DNA and it produces the exact same protein, insulin, that would be coded for in a human. Now remember, you don't have to ever memorize this table. That is not where you need to spend your time. You'll always be provided an amino acid table in some form, and you need to be able to recognize both forms. We'll see the other form in a minute here. So first up, which codons correspond to an amino acid? Remember, 3 letters are used to indicate each amino acid. So each of the 20 amino acids has between 1 and 6 codons. Number 2, what amino acid sequence would be translated from a sequence of codons in a strand of mRNA? So the first 3 bases in the mRNA sequence are the codon for the first amino acid. The next 3 bases are the codon for the second amino acid, and so on. What base sequence in DNA would be transcribed to give the base sequence of a strand of mRNA? So remember that we use the template strand of the DNA and we complementary base pair to it to create that mRNA. All right, this is the other type of amino acid table you might see. In this case, you read from the inside out. So let's try that AUG again. So we're going to start on the inside. Here's the A, and then we work our way out. Here's U, and then finally G. And sure enough, there's our old friend methionine, our start codon. Let's try another one. What amino acid would you get if you had the sequence UAU? Let's try it. We'll start at the center. We'll move out. UAU, and the answer would be tyrosine. Now you should be able to go backwards as well. If you're asked to deduce the codon for a particular amino acid, you should be able to list all of the codons that would code for it. Okay, if you were given codons, you would need to be able to tell me what amino acids that codes for. You might be given a sequence of mRNA that you then need to know. You should draw a line between every 3 so you don't get them mixed up, and then determine what amino acids are coded for. Then you might also be told the sequence of the template strand of DNA. So first, you would have to do complementary base pairing, replacing any T's with U's to get the mRNA strand. And it might be a 2-part question where you then have to use the amino acid table from the mRNA codons to come up with the amino acids. So let's go through the process of translation in a little bit more detail. Translation of an mRNA molecule is done by a repeating cycle of steps. Each cycle results in the addition of one amino acid to the growing polypeptide chain. Once in each cycle, the ribosome is going to move 3 bases along the mRNA, which is one codon. So every cycle, we're going to see this large and small subunit complex shift over in this direction to the right because it's going to go 5' to 3' by 3 bases. Now, before the ribosome even attaches to the mRNA, we are going to need to charge up those tRNAs. And that means we're just going to add that specific amino acid to that specific tRNA. So that's going to happen by a specific enzyme. So each of these tRNAs with their different anticodons is going to have a specific enzyme that only matches in its active site with that particular tRNA and the specific amino acid that matches with it. And so this enzyme is then going to charge up our tRNA, which means the amino acid now is attached to it. So once we have a charged tRNA, it's going to be able to arrive at our ribosome complex, and it is going to move into the first site called the A site conveniently. If the anticodon on this tRNA is complementary to the codon on this mRNA, then those hydrogen bonds will form between complementary base pairs, and this amino acid will then be the next amino acid in this growing polypeptide. The tRNA that's in the P site in the middle will transfer its growing polypeptide to the amino acid, which is in the A site. Picture this is now sitting in this site. Okay, it will form a peptide bond between this amino acid on the end and this next one in the sequence. The tRNA here in the A site is now holding the whole of the growing polypeptide. Then we're going to see a shift of this whole ribosome complex. It's going to move down one codon, 3 bases on the mRNA. That means that these 2 tRNAs that are in the A and P site, those are going to get shifted over. The tRNA that was in the P site is now going to move to the E site. E is for exit. Once the tRNA arrives at the E site, then it can exit, and it can go get recharged up again and transfer another amino acid if another codon was reached that was complementary to the anticodon. Now, there's no more than 2 tRNAs in these 3 binding sites at any given time. So once this tRNA exits, another amino acid is going to be able to enter the A site. That is now going to have an anticodon complementary to the next codon on the mRNA because we've shifted over. The cycle starts all over again. This growing polypeptide chain is going to get passed to the tRNA in the A site. A peptide bond will be formed. The ribosome shifts again. This now uncharged tRNA goes to the exit site and exits, and it continues until a stop codon is reached. Now, it's hard to describe what's happening with a still picture. I highly recommend that you watch a couple of videos on YouTube that show transcription and translation in an animated form where you can see all of the tRNAs moving around. You can see the polypeptide being built. It's going to be much easier to conceptualize and be able to then describe this process if you've actually seen it happen instead of me just trying to indicate things moving. Now, the concept of translation is often going to show up as one of those 7 or 8 mark questions. So make sure you can describe every step in this process, use all the correct vocabulary, talk about the ribosome, the 2 subunits, what the 3 sites are on the large subunit, and then what the process is as we cycle through the addition of each amino acid to this polypeptide. Okay, let's introduce a gene mutation. Now, a gene mutation is just a change to the base sequence of a gene. This could be as small as a single base mutation. Here, there should have been a C, and in this mutation, there's a T instead. Now, sometimes this may code for a different amino acid, and sometimes it doesn't. So, for example, coming back to this slide that has our amino acid chart, say you had UUU, phenylalanine, but there was a mutation, and now instead of UUU, there was a single point mutation that made it UUC. UUC still codes for phenylalanine. So you can see here, that's a silent mutation. Sometimes a mutation has zero effect on the amino acid produced in a polypeptide. This is most often the case when it's the third base that is the mutation. Now, that doesn't mean the third base always makes a difference, right? If you were supposed to have UGG and code for tryptophan, but instead it was UGA, that would be a big problem because it would stop the polypeptide prematurely and could potentially result in a non-functional protein. So a single base mutation might have no effect on the resulting amino acid, a small effect, especially if the amino acid that is the wrong one is similar to the one that it is replacing, or it could have a very radical effect. Let's look at an example where a single base substitution has a radical effect, and that is our example of sickle cell disease, which is the commonest inherited condition in the world. The gene we're going to look at is the gene for the beta-globin polypeptide in hemoglobin. Hemoglobin is the protein that carries oxygen in our red blood cells. Now, our normal red blood cells are shaped like this. They're sort of like discs with these concave indentations on each side. However, the result of this single error, this single mutation of a base, is a sickled red blood cell shaped like a sickle, and there are many medical consequences for this sickling. So most humans have the allele HBA, but if there's a base substitution mutation on the 6th codon of the gene, and where it normally would have read GAG, instead it reads GTG, then we're going to have a new allele formed called HBS. And this mutation can be inherited by offspring if it occurs in a cell of the ovary or testis that later develops into an egg or a sperm. You might think, how can a single base make this massive difference between normal hemoglobin making a round disc-shaped red blood cell and hemoglobin making a sickle-shaped cell? Well, let's take a look at the amino acids produced. Here, where we had GAG, we're going to see that we're going to have the amino acid glutamic acid versus the mutant amino acid valine. Let's take a look at glutamic acid and valine. Now, remember that every amino acid shares this same structure up here that I'm circling. This part then is the R group, which is our variable group. Here's the R group on valine. Now, you might notice some important properties. There is a negative sign on this oxygen. This is a polar R group. It's going to be attracted to water, but it's being replaced by a non-polar R group, which is not going to be attracted to water. And so we're going to see that those amino acids behave very differently as the polypeptide folds into its 3D shape. And so what we're going to see is that the hemoglobin molecules, when they are in tissues with low oxygen concentrations, they link together into chains and form bundles that are rigid enough to distort the red blood cells into a sickle shape. This is the normal hemoglobin. This is sickle hemoglobin. And you can see how vastly different the shape is just based on the properties of this single different R group. Now, the consequences of this sickling is that these sickled red blood cells become trapped in blood capillaries, which blocks them, reduces blood flow, which causes damage to tissues. Now, when sickle cells return to the high oxygen conditions in the lung, the hemoglobin bundles break up, the cells return to their normal shape. But every circuit around the body, they go sickle, normal, normal, which damages the plasma membrane, damages the hemoglobin, and shortens the life of a red blood cell to as little as 4 days. Now remember, a normal red blood cell will last for 3 to 4 months. And so our bone marrow can only produce red blood cells so quickly. And so if these red blood cells are dying after only 4 days, they can't be replaced fast enough to produce enough oxygen-carrying red blood cells. And so therefore anemia develops, that low red blood cell count, and can lead to reduced ability to carry oxygen around the body. And so in conclusion, a small change to a gene can have a very harmful consequence for individuals that inherit that gene. It's not known how often this mutation has occurred, but in some parts of the world, that allele for sickle cell anemia is remarkably common. In parts of East Africa, up to 5% of newborn babies have 2 copies of the allele and therefore develop severe anemia, and another 35% have 1 copy, so they make both normal hemoglobin and the mutant form, and these individuals develop mild anemia. That brings us to the end of the SL content for D1.2. You can turn this off if you're an SL student, but if you're an HL student, we still have more to go. Let's talk about the directionality of transcription and translation. Both transcription and translation are unidirectional processes, meaning they only go in one direction. And we have our one direction picture here to help us remember that. In transcription, each RNA nucleotide is added to the growing strand by linking the phosphate group of the free nucleotide onto the ribose sugar at the end of the growing strand. Transcription is therefore always going to be in a 5' to 3' direction. This is the exact same direction that we saw in DNA replication. It's always going to have another nucleotide added to the 3' end so it can grow in a 5' to 3' direction. Translation, it's the same, folks. 5' to 3'. We're going to see that the ribosome complex is going to move along mRNA codon by codon by codon in a 5' to 3' direction. Remember, DNA replication was also in a 5' to 3' direction. Let's discuss initiation of transcription at the promoter. Now, a promoter is going to be a section of DNA that initiates gene transcription. So it's not going to be coded into a protein, so it's a section of non-coding DNA, typically between 100 and 1,000 bases long, and it's adjacent to genes on chromosomes. The base sequence of a promoter is where RNA polymerase is going to bind along with a variety of proteins that act as transcription factors. Now, the promoter can have either repressor sequences. These sequences allow for transcription factors to bind that prevent the binding of RNA polymerase, and so transcription of the gene is prevented. Alternatively, you could have activator sequences. When transcription factors bind to activator sequences, that then encourages the binding of RNA polymerase, which then leads to transcription occurring. This is going to be a way that cells can change their pattern of gene expression depending on what transcription factors are produced to bind with the promoters. Because remember, we don't want every single protein to be produced at all times. For example, lactase is the enzyme that breaks down lactose, the sugar found in milk. But if you haven't had any milk or any lactose, then there's no reason to produce lactase proteins. That would be wasteful. And so we're going to see that RNA polymerase is not going to be able to bind with this promoter region that's above the lactase enzyme gene until lactose is present in your system. Some transcription factors only bind to the promoter after combining with another molecule. For example, the receptor for testosterone acts as a transcription factor. And if testosterone binds to it, the receptor can then bind to many promoters of genes with roles in male sexual development and activity. And so that can coordinate puberty, for example, by having testosterone trigger the development of many different proteins at the same time. Let's look at some non-coding sequences in DNA that don't code for polypeptides. We'll see that there are base sequences in genomes that don't code for polypeptides, but instead have other functions. Let's look at 5 examples. Number 1, some base sequences get transcribed to produce tRNA. We need to be able to make those tRNAs that then will exit to the cytoplasm, be able to get charged up with amino acids and bring specific amino acids to the ribosome to help build that polypeptide. Similarly, we have base sequences that can be transcribed to produce ribosomal RNA or rRNA. rRNA is what actually makes up the ribosome. A ribosome is made up of rRNA and proteins. Some base sequences are used in the regulation of gene expression, like we just looked at with promoter regions. So the promoters don't actually get turned into polypeptides, but they're still functional. Telomeres are the structures that form the ends of chromosomes in eukaryotes. And we'll see that every time the DNA replicates, those telomeres get just a little bit shorter. Then we're going to see that introns are base sequences that are edited out of mRNA during post-transcriptional modification. Now in prokaryotes, RNA can be translated as soon as it has been transcribed. Let's look at a picture of that. Here we have in a prokaryote both DNA transcription and translation occurring. Let's look at the bacterium E. coli. The DNA is colored pink. mRNA strands are colored green. It's a bit hard to see them, but they're this sort of pea green. And so the mRNA is being produced in this direction. And then in cyan, this teal color, those are ribosomes. And those ribosomes are making polypeptide chains as they move along this mRNA, reading its code. And so we have transcription occurring at the same time as translation. Now in eukaryotes, of course, we cannot have both processes occurring at the same time because transcription occurs in the nucleus and translation occurs in the cytoplasm. Now, one benefit to this is that we can modify the mRNA before it exits the nucleus. We're going to see that newly produced RNA, called pre-RNA, is changed into mature mRNA before it exits the nucleus to be translated. This modification explains the need for that nuclear membrane. We need to make sure that we can modify the mRNA before it encounters any ribosomes. The separation of transcription and translation into separate compartments in eukaryotic cells allows for significant post-transcriptional modification to occur before the mature mRNA exits the nucleus. Let's look at 4 different types of post-transcriptional modification. The first 2. RNA gets modified by adding extra nucleotides to give each end of mRNA a special structure. On the 5' end of the mRNA is a 5' cap. A modified nucleotide is added. Then we're going to see a 3' poly A tail is added. This is poly A because it's made of 1 to 200 adenine nucleotides. So it's adenine, 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 1 to 200 times, and it forms that 3' poly A tail. The purpose of these 2 structures is to stabilize the ends of the mRNA by protecting them from digestion by nuclease enzymes that would otherwise potentially cut it up. They also encourage further modification of the original transcript. So let's look at what those further modifications might be. So here's our 3rd and 4th modifications. Exons and introns. These are coding sequences that are either, if you're an exon, going to be expressed as mature mRNA, which can then exit the nucleus, or introns, these intervening sequences that are not expressed. In fact, they are going to be cut out. They're going to remain in the nucleus and get broken back down into the recycle bin, while exons are going to be able to exit the nucleus as mature mRNA. So RNA transcribed from genes coding for polypeptides contains alternating exons and introns. The introns are removed, digested back into single nucleotides, and therefore recycled. The remaining exons get spliced together to produce an uninterrupted base sequence for translation. We now call this mature mRNA, and it can now be exported from the nucleus for translation by ribosomes in the cytoplasm. Now, what is the purpose of this? Well, this allows for alternative splicing. This is a type of change that allows several variants of a polypeptide to be produced from just one single gene. Here we can see that we have one gene. The primary transcript is the same, but note that we're going to see exon skipping. Here, if we have all 5 exons that are spliced together, we're going to end up with this certain protein. But if when we're excising out the introns, we also took exon 3 out for it and we skipped this exon, we would just be left with exons 1, 2, 4, and 5, which would be a different mRNA and code for a different protein. And this, maybe instead exon 4 was excised along with its neighboring introns, and so we would end up with yet another protein. So this allows for variants of proteins to be made from the same DNA strand. Let's go into just a little more detail about the process of translation itself. Let's look at how it's initiated. Earlier when I described translation, the polypeptide was already in the middle of being produced. So how does this process begin? So we're going to start with an activating enzyme that's going to attach a specific amino acid. In this case, since it's the first one, it must be methionine to an initiator tRNA, and it's going to have the anticodon UAC because there's only one codon that codes for methionine. The initiator tRNA is going to bind to the small subunit of the ribosome. Here you can see that bottom hamburger bun. So tRNA binds. The small ribosomal subunit then binds to the 5' end of the mRNA and starts sliding along it. It's using its UAC anticodon to scan for its complementary start codon AUG. Note that A and U are complementary, A and U are complementary, C and G are complementary. So UAC is the anticodon, which will then bind with the mRNA codon AUG, which we know is our start codon. Okay, when it reaches AUG, the small subunit stops moving. The UAC is linked to the AUG by hydrogen bonds, just like any other complementary base pairing. Then we have the arrival of the large subunit. It's going to bind to the small subunit with the initiator codon right here in the P site. And then we pick up where we talked about before. A tRNA now with an anticodon that's complementary to the next codon is going to enter the A site. The anticodon will complementary base pair with the codon, forming those hydrogen bonds, and we continue the process. The peptide bond is formed as that methionine gets passed over to this amino acid, which is now sitting here in the A site. A peptide bond is formed, and now we have a dipeptide, 2 amino acids. The ribosome complex then shifts one codon over. This now uncharged tRNA gets shifted to the E site, and so it now exits, and the A site is now available for another tRNA to enter. So earlier we talked about 4 ways we can modify mRNA post-transcription. So now let's look at several ways that we can modify polypeptides post-translation so that they can achieve their functional state. These are the main types of change. So first, we're going to remove the amino acid methionine from that 5' end of the polypeptide where it was placed during initiation of translation. Remember, we always start with this methionine, so it's going to be time now to remove it. We can make changes to the side chains of amino acids in the polypeptides, or those R groups. For example, phosphorylation can occur or addition of carbohydrate molecules. This allows us to produce over 100 chemically different amino acids in proteins. Folding of the polypeptide can occur, making intramolecular interactions to stabilize that tertiary 3D structure. For example, disulfide bonds are formed between cysteines that come close together due to folding. We can convert propeptides into mature peptides by removal of a part of the polypeptide chain. 2 or more polypeptides can be combined into quaternary structure. And non-polypeptide components like heme groups can then be combined with a quaternary structure to form a conjugated protein. So this hemoglobin is a great example of both of these latter bullet points. We have quaternary structure because we have 4 polypeptides together, and it's conjugated because we also have these 4 heme groups that together form the hemoglobin protein. Now, as an example, we're going to look at pre-proinsulin and its modification to produce insulin. This is a specific example that IB would like you to be familiar with. Now, don't memorize every single step. This is just an example that demonstrates several of these modifications we just covered, and so it's a nice way to see many of them at work. So let's take a look. The insulin gene is located on chromosome 11 in humans. It's transcribed in those pancreatic beta cells to produce mRNA copies. The mRNA is then translated by ribosomes on the rough endoplasmic reticulum. Remember that the RER is a membrane-bound organelle which is studded with ribosomes. And so those ribosomes are making proteins just like free ribosomes floating around in the cytoplasm, but the proteins are being made directly into the center of those membrane-bound sacs of the RER, the center called the lumen. So the polypeptide that's produced is called pre-proinsulin, and it has 110 amino acids. So as it's made, it passes into the lumen or the center of that RER. Here, a sequence of 24 amino acids called the signal peptide is removed from the N-terminal by a protease enzyme, which recognizes 3 adjacent alanines in the amino acid structure. A chain of 86 amino acids remains, which is called proinsulin. So we lost the pre by cutting off 24 amino acids. So going back to this last slide, remember we're going to see this bullet then, conversion of propeptides into mature peptides by removal of a part of the polypeptide chain. Next, proinsulin becomes folded with 3 disulfide bonds stabilizing the tertiary structure. Here is this bullet point, folding the polypeptide, making those intramolecular interactions to stabilize the tertiary structure. Next, peptide bonds are broken at 2 positions in this proinsulin we now have by proteases, which are enzymes that recognize pairs of positively charged amino acids, lysine and arginine specifically, but again, don't memorize this level of detail. This causes a sequence of 33 amino acids to be excised and leaves 2 separate chains. The A chain now has 21 amino acids, and the B chain initially has 32 amino acids. They're still held together by those disulfide bonds made earlier up here where we looked at folding of proteins. A pair of amino acids is then removed from the C terminal of the B chain by a protease to yield mature insulin with a total of 51 amino acids. And this is what it looks like. Here you can see 1 chain, 2 chains, and here are those disulfide bridges holding them together. And this is the 3D structure of insulin, our hormone that is going to indicate to cells when blood glucose levels get too high to take some of that glucose out of the blood. Lastly, recycling of amino acids by proteasomes. A genome is all of your genes that are present in your body, and they're in every somatic cell is your entire genome, your whole collection of genes. Your proteome is your entire collection of proteins. And to sustain a functional proteome, that's going to require constant protein breakdown and synthesis. Most of the proteins produced by translation have a relatively short life in the cell. There are many reasons for this. Number 1, the cell's activities may change and a protein is no longer needed. An example of this is when a cell moves from one stage in the cell cycle to the next one. So looking back on our example, we know that DNA polymerase III is going to be placing DNA nucleotides during DNA replication. But that's only one point in a cell cycle that DNA replication has to occur. If we've moved past that stage, we don't want to keep producing DNA polymerase III. That's wasteful. And so it's going to be important that we only produce the proteins needed at any given time for that cell. The structure of many proteins is quite easily changed by free radicals or other reactive chemicals. And if this happens, the protein can no longer carry out its function. So we don't want that protein to stick around. The protein could become misfolded or denatured in other ways. So proteins that no longer have a function or are unable to carry out their function are broken down by proteasomes. Proteasomes break proteins down into short chains of amino acids. Further digestion of these oligopeptides in the cytoplasm breaks them down completely back into their building blocks called amino acids. Oligo just is a prefix that means few. So we're just saying it breaks it down into just a few amino acids, and then now they're going to get fully broken down into individual amino acids that can be recycled and reused for synthesis of new proteins. The proteasome thus removes functionless and damaged proteins from the cell, but it also recycles amino acids. That takes us to the end of D1.2. Good luck out there.